Cool. All right. Hi, everybody. There are cookies and coffee and hot chocolate in the back if you didn't get some. If you didn't know already, it's going to be a very busy start of the semester in EEB. We have our departmental seminar series, which is kicking off today, that I'm very excited about. But starting next week, we also have seminars starting for the director of MPOBI and a tenured position in the Ecology and Evolution of Infectious Disease. So there will be 4 candidates on campus, and please do engage with them at the level that we always engage with these candidates. We want feedback from the entire community on these candidates, faculty, staff, grad students, postdocs. Please do come to their talks and engage in their science, because we're recruiting a colleague for, on average the next 30 years, but we also all know John Vandermeer, so it could be quite some time for this person to be in our department. Soon after that, we start the interview process for an assistant professor in global change biology. Again, it's really exciting that we're hiring in the ecology and evolution of infectious disease when we all know what's happening with NIH and the CDC and other organizations. It's also exciting that we're advertising to hire in global change biology. We're one of 2 R1 universities in the country that are advertising for a tenure track position in global change biology. I think both of those jobs send a really important signal about what this department cares about, what this university cares about, and that we're willing to continue to fund research in areas that are critical for people and the planet. So please do engage with these folks. And I'll turn it over. This one works.
Hey everyone. So it is my pleasure to introduce our Thursday seminar speaker, Dr. Lynette Strickland. Lynette received her bachelor's degree from Texas A&M, go see Aggies, and went on to complete her PhD on tortoise beetles at the University of Illinois Urbana-Champaign, advised by Carla Caceres. As part of her graduate work, Lynette also conducted research at the Smithsonian Tropical Research Institute with Don Windsor. She then completed postdoc fellowships at Texas A&M Corpus Christi with Dave Portnoy and at the University of Memphis with Dwayne McKenna as an NSF postdoc fellow. Hello? Okay. And then in 2023, Lynette opened the Cassidines Ecology and Evolutionary Genomics Lab at Boston University. Her lab seeks to understand the evolutionary origins and maintenance of variation using color polymorphisms in neotropical beetle communities by integrating natural history, ecology, behavior, and genomics. Recently, her work has been published in Nature Communications, Earth and Environment, Proceedings of the Royal Society B, and Ecology and Evolution. In addition to this amazing research program, Lynette is a champion of building equitable, anti-racist labs and institutions through inclusive mentorship, community engagement, and international collaboration. She's also a board member of Black in Genetics, a nonprofit that amplifies and promotes the voices of Black geneticists within the US and globally. Her advocacy has remained consistent throughout her career. As a graduate student, Lynette authored work on sustainably increasing diversity in science, and that work was published in Science and Nature, Ecology and Evolution. I first met Lynette when she gave a talk at the University of Georgia in 2023. We had a fantastic dinner when I was a graduate student, and I was immediately fascinated by her work on these shiny beetles. I also thought she was an incredibly cool person, and I know by the end of her talk today, you'll feel the same way if you don't already. So please join me in welcoming Dr. Lynette Strickland as she speaks on color, cold, and Cassidines and integrative approaches to studying adaptive evolution in tortoise beetles.
That was such a fantastic introduction. I feel like we can just go home from here. Thank you so much. And thank you all so much for inviting me. I've had a really fantastic day talking science, talking life with all of the folks that I've met with today. So I'm really excited now to share about a lot of the things I alluded to throughout the day. So I'll go ahead and get started, maybe. So just a quick outline. So I'm going to talk about a lot of my dissertation and postdoc work, which has looked at color variation in a particular group of beetles. So working to connect genotypic variation with phenotypic variation. Okay, sorry. And thinking about color variation. Thank you. that better? Maybe slightly, ever so slightly. I'll face this way only.
Is the other mic still?
Yeah, maybe it's still on.
Oh, yeah, there we go.
Okay, awesome. And then I'll end up by talking about some new directions that my lab has gone in over the past year or so, thinking about more complex phenotypes. So if we can still connect genomic variation with more complex phenotypes, specifically in thinking about color variation. So beetles, why am I fascinated by beetles? They are incredible. They are a really incredible order, or order Coleoptera. There are over 350,000 described species of beetles, meaning 1 in every 4 metazoans is a beetle, which is an astounding amount of species diversity when you think about it. So thinking about how these organisms have evolved to be so diverse is one of the things that we can do with this system. They're also really old. So they evolved during the Permian, so they have a 300 million year history, have survived multiple mass extinctions, and have these really cool, often tight associations with host plants or with other food resources. So they provide a really great opportunity to ask questions about coevolution. And they're also actually relatively understudied. So when we look at specifically thinking about genomics, so the number of beetles that have a complete and annotated genome, it is actually highly underrepresented compared to other insect systems. So they provide this really cool system to ask a lot of questions about the maintenance of diversity, the maintenance of variation, but also allows us to connect this with genomic variation. So thinking about what strategies do beetles use to enhance their own survival? What has allowed for this incredible success of the order Coleoptera, or beetles? So my work looks at specifically, can we get a sense of what genomic variation is sort of shaping and maintaining the phenotypic variation that we see in a particular group of beetles? And that group of beetles being tortoise beetles. So I'm enamored by tortoise beetles. I've been working with tortoise beetles for the past decade or so. They have these really cool, often these extended elytral margins, right? And so whenever they feel threatened or under attack, they will often pull everything in under those elytra, and that's where they get the name tortoise beetles. As larvae, they have this excellent fringe, and so that is essentially different molts and their own poop that they collect and keep on their back. And as siblings, they will often wave those poop shields around. We literally call them fecal shields. And this is a form of defense against predators because nobody wants to eat things covered in poop. So they have a bunch of really cool, unique strategies. And to me, I just think they're a very charismatic group of organisms to ask the questions that I'm interested in. So I've been focused, a lot of my work has been focused on this particular species here, and this is most of what I'll talk about today, but like I said, I'll get into some new directions my lab is taking. So this is Cheilomorpha alternans. This is a polymorphic neotropical tortoise beetle. So here I'm showing you the different discrete color morphs here, and then we also have like some more continuous variation that we've seen in this red and black morph. So when I first saw this beetle, I was immediately intrigued by thinking about both the genomic mechanisms, but also the ecological mechanisms that are maintaining this color variation. And so I'll talk about the work we've been doing in trying to get at the genomic basis of color variation, but also some of the work we've been doing in trying to understand what ecological or environmental factors maintain this variation. So first I wanted to get a sense of do these beetles actually co-occur in their natural populations, right? Do we see populations harboring more than one phenotype? And so this was a study of 28 different field sites across the Isthmus of Panama. It was really fun. It was essentially just going out to all of these different locations, surveying them, and counting the different phenotypes that have co-occurred there. So our little cargo ship is lining up with the opening of the Panama Canal just to orient us. And so each circle represents a collection site, and the site of that circle is the number of beetles that were sampled there. So just a couple things I want to point out is that we have two phenotypes that are very widely distributed across the Isthmus of Panama. So they occur from the western end to the eastern end of Panama. And then we have two phenotypes that do seem to have some geographic partitioning. At least these two phenotypes, our red and black morph and our all red morph, never co-occur in the same natural geographic population. So I feel like this does provide a foundation for some interesting questions about environmentally what might be maintaining this. But what we can see is that almost every single one of our populations harbors at least two and often three of these different color pattern phenotypes. So this does provide a foundation to ask, how is this inherited? How are these phenotypes inherited? And how are they maintained ecologically and genomically? And so that's what I'm going to walk us through for the main portion of this talk. So it's thinking about how these color polymorphisms are inherited, looking at the work that we've done genomically in getting at population differentiation, and ultimately what I'm trying to get at is what is the genomic basis of color, and then some work looking at sexual selection, mate choice, and predation as potential factors maintaining this color variation. So to answer that first question, I really got in tune with my inner Mendel. And this involved over 100 different controlled crosses. And so essentially what we would do are these controlled breeding crosses where we take males and females of known phenotypes, we mate them in a controlled setting, and we count all of the resulting offspring that come from that particular cross, and we assign the phenotype of the offspring that came from that cross. And so this is just three examples of different crosses, the phenotypes of metallic or variguenses of each different cross, the total amount of offspring that came from that cross. So as you can see, this was a lot of beetles to raise up, and the observed phenotypic ratio from that particular cross. Does anyone notice anything about these observed ratios? Yeah. They are not significantly different from what the observed ratios we would expect to see under these classic Mendelian scenarios of a homozygous dominant individual with a homozygous recessive individual, a heterozygote with a homozygous recessive individual, or two heterozygotes. And so that's actually one of the things we noticed consistently about all of these different phenotypes that we were mating in these controlled crosses, were that we would consistently get these 3-to-1, 1-to-1, or 1-to-0 phenotypic ratios in the offspring. So this was incredibly cool, right? To see a polymorphism that there was a little bit of variation with that black and red one. If anyone has questions, we can chat about it. But at least these 4 discrete phenotypes, we can accurately predict based on their genotypes, right? What the phenotypic ratio of the offspring will look like, with that metallic individual being this homozygous recessive phenotype. And so when we put this entire data set together, what we see is that we have these 4 segregating alleles that has a clear dominance hierarchy, and that metallic phenotype is only expressed in the homozygous recessive state. Now this was really cool because we also found that it was our most abundant phenotype occurring in every single population that I sampled across Panama. And so there's actually been some other work looking at insect polymorphisms that have shown that the most recessive phenotype is also the most abundant phenotype. But I think it provides a foundation for asking a lot of really interesting questions as to what exactly is making this individual perhaps more successful in different populations. And so I try to get at that a little bit, and we'll talk about that through the rest of this talk. But so this was really fun to see, and it provided a foundation to do a lot of really cool work now that we had this foundation. And so next I'll go into thinking about genetic and genomic variation, right? So I'm very interested in understanding, like, within a population, we know that multiple phenotypes are occurring, but what's the level of genetic differentiation within and between populations? And also what I would really like to get at is what is the actual genomic basis of color variation in this species? So I'll talk a little bit about some tools that I use to get at some of these questions. A big tool that my lab uses is called RAD sequencing. Rad sequencing is a fantastic tool. It essentially takes restriction enzymes. It cuts at particular locations across the genome. And so you can take these snippets of DNA or snippets of your genome, and you can ask really great questions about like population structure, levels of genome, levels of hybridization. I just want to point out this is a fabulous, fabulous resource. I use it a lot. I recommend it for like grad students very often choosing a population genomics approach, which is where this figure is from. But one of the things to note about rad sequencing is that you're taking a very small portion of the genome, often like 5% of the genome. And so it allows you to ask really nice questions about population structure, and it's really great for working with systems that have no previous genetic or genomic information because you don't need a genome. Right, but it is limiting in terms of the breadth of the genome that you're sampling. So that's one of the trade-offs here. But we use it a lot to ask questions about population structure, which is what it's a really great tool for. And so this first study was just trying to get at what's the level of genetic differentiation among color phenotypes within the same geographic variation. So to answer this question, I went to one of my field sites, Santa Rita Ridge Road here in Panama, collected individuals of each of those phenotypes, and then performed a rad sequencing study. I do want to point out, driving to a lot of my sites is often really beautiful and fun, and it's a great time. The beetles are specialists on morning glories, which I got to chat with a lot of people about today. Morning glories grow really well in disturbed habitats. So I actually often end up in trash dumps are a great place for morning glories, and as such a great place for the beetles. So while I'm driving to these beautiful locations, I end up sampling here. But it's still a really great time. And so from this data, basically what we see, when we get our genome scans back, we analyze the different SNPs that we're getting from this data, and we're looking at how differentiated these SNPs are between these different phenotypes. And so in this particular case, I'm using phenotype as a proxy for population. So normally you would do this with different geographic populations. And again, this is just to get a sense of how different they are when they're occurring in the same geographic population. And so this is a discriminant analysis of principal components. And so what we can see is that for the most part, there's not a lot of genetic differentiation among phenotypes that occur in the same population. So when we look at our FST values, those values that are giving us a sense of how much genetic differentiation there is, measured from 0 to 1, we see that these are pretty low. These are very low values. And so from this, I would hypothesize that within a single population, phenotypes are interacting with one another, they're mating with one another, and that we see these different, again, we see these different phenotypic ratios in a single egg clutch. So now that we had this sense of how related phenotypes are within a single population, I wanted to look again between geographic populations and then also try and get at the genomic basis, ideally at the same time. Didn't quite work out. But so another technique we use a lot is pooled sequencing. So pooled sequencing is another essentially form of reduced representation of the genome. In this case, you pool all of your individuals into a single library. And so while you lose that individual level of information, you gain a much broader breadth of coverage and often better depth of coverage. So there are two different tools for getting at different types of questions, population structure versus finding like divergent areas of the genome. And so this pool sequencing approach is what we use for this next study, which is trying to assess what's the level of genetic variation across these geographic populations, and can we find divergent areas of the genome that are associated with phenotype. And so for this, I had 4 distinct populations, and I focused on just 2 of the color pattern phenotypes, that red and black and the metallic one, and sampled the individuals of both phenotypes across 4 different geographic populations. And so when we look at, I just want to say, the analysis of this is being led by an awesome postdoc in the lab, Dr. Sam Kubica, who's been really fantastic in like sticking with all of the bioinformatics analysis for this project. So again, looking at FST just within a geographic population, we once again see low levels of genetic differentiation, indicating that within a population, individuals are interacting, mating with one another. When we look at between our different populations, so here each symbol is representing a different geographic population, and we have each beetle representing its particular phenotype. We see that between geographic populations, there is a little bit more genetic structuring. I will note these individuals up here are only separated by 40 or so kilometers, so they're much closer together. When we go a little bit further out, when individuals are separated by 80 kilometers, understandably, we see a greater degree of genetic differentiation. And when we go even further out, getting over 200 kilometers, we see a greater degree of genetic differentiation. So within a population, individuals are likely breeding and interacting with one another. Between populations, we see more restricted gene flow. Now, the other really cool thing that you can do with pool sequencing data and with an experimental design set up like this is that now we can now pool all of these populations into just two populations, just the metallic and just the rufipennis phenotype, and then account for that geographic variation. So essentially get rid of that geographic variation in order to find areas of the genome that are accounting for the variation just between these two phenotypes. And for this, we do need a genome. So I'll show, our genome is not great, right? So it's about 100,000 scaffolds. So we just got some PacBio Hi-Fi data, hoping to clean this up a bit. But when we align this pool sequencing data to this not very great genome, we still see this massive peak right here, right? So this is essentially our candidate region for an area of the genome that is contributing to the divergence between these two phenotypes. So my hope is that when we annotate this, when we go look in this genomic region, we find genes that are associated with the different color variants. So that's our next steps for this project, is really figuring out what's in this particular genomic region. But it was really exciting that we found this even with like not the most contiguous of genomes. Okay, so next I'm going to talk about the work that we've done with sexual selection, mate choice, and predation in this system. So thinking about ecologically what might be contributing to the maintenance of these polymorphisms. And so what was really nice about this, answering this first question, do color pattern phenotypes mate preferentially? Do we see either assortative or disassortative mating between these color pattern phenotypes? Was that I could rely on all of that work that we had established at the foundation. The fact that we could have these controlled breeding studies meant that I could set up what I called a triad. So a female of known genotype with two males of known genotypes. And I knew, because of all of the work that we had done previously, what the expected phenotypic ratio of the offspring would be if she's mating assortatively, disassortatively, or random. So the hypothesis under the disassortative mating, so if the female prefers to mate with a color pattern phenotype different than her own, which might be a mechanism that maintains variation in populations, then I would expect that in this particular case, this female's egg clutches are predominantly reflective of a 1-to-1 ratio of rufipennis to metallic offspring. I could generate the same hypothesis for if this particular female prefers to mate assortatively, then most of her egg clutches should produce offspring whose phenotypic ratios match a 1-to-0 ratio of all metallic offspring. And I could generate this for if the individuals are mating randomly, right? So this would give me a sense of, do individuals prefer to mate with their own color pattern phenotype? Do they prefer to mate with a different color pattern phenotype? Or do they mate entirely randomly for color? And so to walk through this data set, basically what I'll show you is our female phenotype here with the different male phenotypic pairings across the top. And so either assortative or disassortative. And so for here, I just had 7 triad types, so 7 different combinations of males and females, just under 500 egg clutches, and again, thousands of offspring. So these were always like tons of time in the basement of a lab, rearing little beetles and bringing them their leaves. But it was a great time. And so here what I'll be showing you is if there is red coloration, that's the proportion of egg clutches that match what we would expect to see under assortative mating. Gold is showing the proportion of egg clutches that match what we would expect to see under disassortative, and green is showing the proportion of clutches that would match what we would expect to see under random mating. So in this particular case, we can see that this female, half of the egg clutches she produced match what we would expect to see under random mating, about a quarter matched disassortative mating, and another quarter matched assortative mating. And so when we take this whole data set together, the main thing I want you to pull out of this is that for the most part, 50% or more of all of the egg clutches that females are producing from this experiment match what we would expect to see under random mating. So we don't see consistently individuals choosing to mate with their own color pattern phenotype or choosing to mate with a different color pattern phenotype, but at least for color, they're mating randomly. Now, I could also look at not just triad types, but pairings. Either assortative pairings, so both the male and female are the same color pattern phenotype, regardless of what kind, or disassortative pairings. So they're different color pattern phenotypes, again, regardless of what time or what kind. And I could track their offspring development through time to get a sense of whether offspring fitness differs based on whether the parents are assortative or disassortative. And so this is exactly what I did. So I essentially tracked each one of these egg clutches into emergence from the egg clutches through their larval developmental stage, into their pupal stage, and into adulthood, and compared this between assortative and disassortative pairings. And this is what we see. So again, gold is showing us disassortative pairings, and red is showing us assortative pairings. So what we see is that offspring from disassortative pairings, so offsprings whose parents are of different color pattern phenotypes, are twice as likely to survive through pupation and into adulthood than those individuals who come from assortative pairings. Now, this I thought was really cool because it is a drastic difference between those offspring of assortative pair types and disassortative pair types. And this was regardless of what kind of combination of disassortative or assortative. It was only whether they were different or the same. Now, the other thing I could do with this data set is not just track offspring survival, so the overall proportion of offspring that survive, but differences in fecundity. So differences in the number of eggs laid by females from each one of my pairings. And so again, red is showing us assortative and gold is showing us disassortative. And what this is showing us is that females from assortative pairings lay more eggs per egg clutch than females from disassortative pairings. That's what I'm saying here. So this led me to a really cool, and what I think is happening here, potential reproductive compensation mechanism. So we see this actually a lot in birds, so we see this in mallards, in finches, where females of suboptimal pairings actually invest more in the resources of those offspring. And so this is my current hypothesis for what may be going on with this data in this species here. My grad student who joined the lab about 1.5 years ago was really interested in following up on this work, and so that is one of the things she did. And she was really interested in like what might be the mechanism underlying this, like how exactly are females compensating for this reduced larval survival with higher fecundity. And so what she looked at was a bunch of video data of mating events between different pair types. And what we see is that assortative pairs spend a much longer mating. So this is my PhD student, Sarah. So they spend way more time actually in copulation than those pairs that are disassortative. Sorry, I thought that was a question. Which we think is a potential mechanism for producing this increased fecundity, so producing more eggs per egg clutch, because it increases the amount of sperm that gets transferred overall. She teased this apart by the different like phenotypic pairings, right? So here we're seeing the different combinations of assortative beetles and the different combinations of disassortative beetles, and this pattern holds regardless of what the exact phenotypic pairing is. So this is our current hypothesis for at least what might be happening in mate choice in this species, is that perhaps it's some sort of bet hedging strategy, and that's why we don't see disassortative mating biased populations, even though those offspring from disassociative pairs are so much more likely to survive, and that there's some sort of reduced compensation with more eggs per egg clutch. So that's some really fun work we've done with thinking about how mate choice and mate preference may, or in this case, may not be maintaining this color polymorphism that I've been so interested in. Next, I'm going to take you through some work that we've done with predation. So these are very brightly colored insects, right? So this very quickly led me to hypothesize that they're aposematic, that they are sequestering toxins from the host plants that they consume, that they are chemically defended, and then that they are advertising this chemical defense to predators with this really bright, beautiful coloration. And so to do this, I essentially did a series of invertebrate predator bioassays, which was also a really fun project. And so the null here being that phenotypes experience equal or random amounts of predation, and the alternative hypothesis being that some phenotypes are better defended than other phenotypes from predation. And so this study involved adult individuals. It would be really fun to do this at the larval stage. But so just under 200 trials, I used these phenotypes of beetles here, and these are my invertebrate predators that I used. So Azteca chartifex, these carton nest ants, they form these really beautiful teardrop-shaped nests from the tops of trees. Trichonephila clavi, sort of the orb-weaving spider, which was really fun to work with, and mantises. So these are all fierce invertebrate predators that are dominant throughout neotropical communities and would occur in all of the same populations or microhabitats that I would also find the beetles. So that is one of the reasons why I chose these 3 species. The other reason I chose these 3 species is that they all have these very distinct rejection responses. So for mantises, one of the responses I was looking for was, of course, consumption. So do they eat a beetle that's presented to them? Or rejection? Do they show a rejection response to a beetle presented? So here you see a strike, and then a very quick flicking away. And so those forelimbs of mantises, that is actually how they taste potential prey items, right? So you see this drastic strike and this sort of touching and tasting of it, and this really drastic release of what they do not consider to be a prey item, right? So I was looking for either consumption or rejection responses in my mantises. Orb weaver spiders, so they form these really beautiful, delicate, very intricate webs, and they rely on prey items getting stuck in the web to consume them. And so they have to keep these webs free of debris, because sticks or leaves in the middle of what looks like nothing automatically makes their web look very conspicuous. So they do this behavior where they will clear debris out of their web. So if something gets stuck there and they sample it, it's not a prey item, they essentially cut around it from their web, and then they release that thing from the web. So this was a rejection response that I was looking for in my spiders. Nope. Or I was looking to see a consumption response. Do they actually view this organism as a potential prey item, and do they go on to consume it? So this is an example here of an individual consuming a metallic beetle. And then finally with my ants, so this was the one predator I did not want to bring back into the lab because it would have been painful. So I brought the experiment to them. So because they have this really, essentially this home base with their tree, I could set up these platforms on the base of a tree where I found a carton nest ant, and I would bait the platforms with tuna over 2 weeks prior to the start of all of my trials, so that the ants will get used to coming to this platform. And then prior to the start of trials, I would assign each tree a particular phenotype. And so essentially I would go and I would put a beetle on that platform and look at their response. And so the consumption response I was looking for was removing the beetle from the platform, taking it to the base of the tree and carrying it up to the tree into the nest, presumably where it's used as a food source for developing larvae. So this is the proxy that I'm using for consumption. I can't actually see them consuming the beetle, but they also have this very distinct rejection response where they take that particular item and they take it to the edge of the platform off the base of the tree and they drop it off. And they would do this with they really started to see this platform as part of the home base. And so they would do this with leaves, sticks, debris, just things that were unwanted on the platform. And so in this case, you saw they're treating this beetle essentially as trash, as debris, right? It gets removed from the platform. And so when we look at all of this data together, this consumption and rejection data, and we're looking at this question, do predators experience different levels of predation? The answer is, well, it depends. It depends on which predator. So what we see from the spiders is that they pretty readily and happily consumed all of the beetles, regardless of phenotype. When we look at the mantises, we see that they did not consume any beetle. There was not a single mantis in this study that consumed any of the beetles, regardless of phenotype. So again, both indiscriminate in consuming or non-consuming. The ants' response was really interesting because here we do see a differential response based on phenotype. So I am going to delve into the ant response a little bit more. So we can separate this by the different trials I did, right? So I did 10 trials with the same ant nest. This was to test for learning, so if they would learn to avoid a particular prey item. So what we see over 10 trials for this particular phenotype is that it's kind of all over the place. Sometimes they would eat the beetle, sometimes they wouldn't, sometimes some colonies would. So overall, there was no particular trend here. We do see a slightly downward trend. However, when we compare that with our all red morph, what we see is that 2 of the 3 ant colonies did consume the beetle in the first 3 trials. And then after that, not a subsequent colony consumed the beetle. So this is a pattern that we would expect to see under learned avoidance behavior. So the predator samples the individual prey item a couple of times, learns of its distastefulness, and in all subsequent encounters avoids that potential item. And the metallic phenotype was maybe one of the more interesting. There was not a single ant colony in any of the trials that consumed any individual of the metallic phenotype. So we do see a little bit of differences from our ant predators based on phenotype. And so one of the things I'm really interested in is how predators might have detected these differences. So potentially looking into cuticular hydrocarbons is one of the things I'm really interested in going from here. Not going to chat a ton about that now, but if anyone has any ideas or thoughts about that, I would love to chat more about what might be differentiating these 3 phenotypes for our invertebrate predators. Okay, so to wrap up this section of the talk, thinking about color variation in cheilomorpha alternans, what might be maintaining it. We talked a little bit about genetic and genomic differentiation, still trying to get at that level of the genomic basis of color, and different avenues that I've gone in thinking about what ecologically might be maintaining these color polymorphisms across populations. So I wanted to take a little bit of time and just chat about some of the things my lab is working on now. So we're still thinking about what's happening in this system and all of the different interesting questions it sparked. And color is really great because it provides a relatively simple phenotype by which we can ask a lot of interesting ecological questions. Lately, I've gotten into thinking about a bit more complex phenotypes, and can we still connect this interspecific variation in a more complex phenotype with genomic variation? And so this started as a collaboration in my NSF postdoc at the University of Memphis with Dr. Lee Boardman, who is an insect physiologist and all-around super cool human. And basically we wanted to do some bioassays with the beetles that I was keeping in her lab, that she let me keep in her lab, thankfully. And so one of the things we did was this bioassay, this physiological bioassay called chill coma recovery. And it's a really nice, simple assay. It's actually really fun and easy to do, especially in undergraduate classrooms, where essentially what you do is you expose an insect to 0 degrees Celsius. They enter this coma state as a response to the drop in temperature. And then once you remove them from 0 degrees Celsius, put them back at room temperature, you essentially just record the amount of time it takes them to recover from this coma. That's called the chill coma recovery time. And so you can look at differences in this chill coma recovery time between species, between individuals raised in different populations, and you can get a sense of those individuals that recover faster, those species that recover faster, are more tolerant to colder temperatures. Now, what I wanted to ask was, could we look at this across a neotropical community? Could we see if there's variation in chill coma recovery time for neotropical beetles? And essentially, can we find differences in gene expression patterns that are associated with the ability to recover more quickly or less quickly? No. Okay, so this is work that has been led by an amazing, amazing intern who is, hopefully, cross your fingers for me, joining my lab this coming fall. So she's been working down in Panama for the past year, doing a bunch of different projects. But one of the things that she did was she measured chill coma recovery time across 16 to 20 different species of tortoise beetles, all found in Panama. She went out collecting, brought them back, ran the bioassays, and she did this with over 500 individuals. So a really cool, incredible data set. And one of the things that I think is awesome from this is the amount of variation that you see in the chill coma recovery time across neotropical beetles, right? All occurring in the same environment. And what we see is that there are some individuals that are fast recoverers. So Cheliomorpha alternans, who we've been talking about, Corydotella sexpunctata, who we'll talk about a little bit more in a second. This is an incredible beetle. But we also see that there are some slow recoverers, including some species in the same genus, right? Are much slower recoverers than other species in the same genus. So this has really got me interested in environmentally, phylogenetically, thinking about evolutionary history, what is contributing to this variation. But lucky for me, there are some species of tortoise beetle that have been found in every population where I've lived, which has been great. And so that is the golden tortoise beetle here, Corydotella sexpunctata, and the mottled tortoise beetle. So they both have a really wide distribution. The golden tortoise beetle has the widest distribution of almost any tortoise beetle that has been described thus far, ranging from southern Canada to Argentina. So it's dealing with incredible environmental variation. And in each one of these populations, either I or an intern or undergrad performed chill coma recovery time. And one of the cool things we see is that by geographic population, you kind of see exactly what you would expect, where those populations in Boston recover faster from the chill coma, so they're more tolerant of that colder climate. But also in every single one of these populations, the golden tortoise beetle is a faster recoverer consistently than the mottled tortoise beetle. And so this has led me and members of the lab to think about genomically what might be enabling this. And so there's a really amazing undergrad researcher in the lab, Maya, who is really interested in essentially taking beetles from these two species out of their coma at different times, so sampling them at 0 minutes, 5 minutes, 9 minutes, et cetera, and extracting their RNA and doing a differential gene expression study to essentially see if we can find genes that are associated with faster recoverers, and see ultimately if we can get a sense of genomically what might be contributing to the variation that we see in this more complex phenotype of chill coma recovery. So to sort of conclude and bring everything together, we talked primarily about a lot of the work that me and my lab have done in thinking about what's maintaining color variation in a polymorphic beetle, trying to get at the genomic basis and some fitness consequences of predation, and then some new directions my lab is taking to think about this variation in chill coma recovery, so looking at interspecific variation. I have to thank my incredible lab. It's been so fun and has really reinvigorated my love for what I do and the questions I ask by having students who ask their own fun, exciting questions that I get to help them on. My incredible mentors, collaborators, funding sources from the Smithsonian and the NSF. And just quickly, I want to say this is in loving memory to my best friend who recently passed and who always thought it was equal parts very weird and very cool that I decided to spend my life studying beetles. So with that, I would be happy to take any questions. Yeah.
They really didn't show much of a preference except for the 1/3 of not the things of the mentality. Is there any connection between the biases, the biases?
Yeah, so like connecting that differential survival at the larval stage with that differential success at the adult stage. So here's my like big picture hypothesis for this that I was like my next NSF grant. I am hypothesizing that some phenotypes and genotypes are better able at dealing with sequestering secondary metabolites from their host plant. So that those like maybe get hit really hard at the larval stage, so they don't survive well at the larval stage, but once they make it to adulthood, all those metallic individuals potentially have just more higher concentrations of all of these toxic chemicals that make them better defended at the adult stage. That's my hypothesis for that. That's a great question though. Yeah. I'll come this way.
I have a question about the reception. Like, if you have any idea of the compounds in the morphing flores that you could be using as those metabolism like themselves, and why does it not affect spiders if it affects mantises?
This is a great question. Do you want to come answer it in the lab? So the question was, what do I think they might be sequestering from their host plants, and why did it not seem to affect spiders? I don't know what it is they're sequestering. There have been some studies with GCMS on their main host plant in Panama, Marimia umbellata, and it just seems to be chock full of like all kinds of alkaloids. So potentially things in that class, but I'm like, my chemistry is bad. So I would love to know, essentially do GCMS studies of the plant and the beetle to see if we're finding the same thing in both of them. My hypothesis for this particular spider in particular is they have an injection style of consumption. So actually the part of the beetle that they leave behind in every study are the wings, so the elytra, the color-bearing portion, and the pronotum, the head. And so essentially what they do is they inject in the soft underside, they liquefy everything and suck it back up. So part of me wonders if they just they can't detect maybe what the ants and the mantises were able to detect with like cuticular hydrocarbons or something. That's just a hypothesis. Yeah.
Okay, sorry, brief follow-up.
Yeah.
I was just looking up where morning glories get their compounds from, and it says that there's a fungal neutralists who provide like ergic alkaloids, and so you think that maybe there's like a multi-trophic like thing happening?
Totally, totes. There's this really cool lab in Germany at Max Planck that works more with like gut endosymbiont and like consuming the plant and stuff like that. Yeah, so they do a bunch of cool work and thinking about all of these different trophic interactions, thinking more about like fungal communities. Yeah, yeah.
As a beetle, how do you make a shiny gold spot? How many options do you have?
And how many options do you have to make a what?
The red pigments.
The red pigments. Okay, so here's what we know about how they are gold from a closely related beetle species. So within the elytra, there are these really thin chitinous, so they're made of chitin, they're elytra, these really thin chitinous layers that are laid down. And so it's differences in the thickness of space between these layers that basically when the light hits it, reflects in a certain way so that we see gold. That's like my best explanation from it. So that's what we know so far. I've tried so hard to take these beetle wings, cut them in half, look at them with a TEM to see if, right, for instance, where that gold stripe is and that red stripe is, if you see differences in like chitin thickness layers. I cannot find anyone who can figure this out, who can help me figure this out. To make the red coloration is carotenoids. So we've been able to look at that with GCMS. And so it seems to be primarily carotenoids for that red pigment.
And are they synthesizing that, or are they getting that from their food?
Oh, that's a great question. My guess is that they would be getting it from their food. So I don't think I have any pictures, but when they first eclose from their pupal form, they are a very, very pale, like orangey yellow type color. And it takes 2 to 3 weeks for that full, bright, beautiful red, as well as that full metallic coloration to actually form. So there is some delayment in the development of that coloration. Yeah. Any other questions? Oh yeah, Haley.
That was awesome. In your map of Panama, where you kind of have the partitioning of some of the color ones, have you thought of that or explored if there's any overlap in like color space with other spicy red and black things like the ladybird beetles and assassin bugs look the same as those guys in that area?
We're just so thinking on the same wavelength. Thought about, yes. Done, no. But yeah, that's one of the things I've wondered basically is like if you track other, like not even just beetles, but like other insects that are aposematic or signaling in some way, do you see, for instance, like there's more all red individuals down where we see mostly that all red individual? Do you see like if we move further south into Colombia and further south, do we see that there's more of that red and black striped individual, which we saw like really high numbers of in that portion of Panama? So yeah, thinking about if there's just this giant mimicry-ish complex going on where everyone's just benefiting from having the same sort of signals is definitely something I thought about. I have no idea, though. Cool question. Yeah.
How or do you think that the beetles are aware when they're mating assortatively versus disassortatively? Because they don't have mirrors, do they know inherently I'm a metallic beetle and I'm choosing this mate? Because I know it's random mating, but they seem to increase or decrease their copulation times. So that would
Yeah.
imply that they know something about their
Yeah, that is such a good question. So I don't know if it's necessarily that they know like I'm a metallic individual and you're a metallic individual, but I have done some preliminary GCMS work with different phenotypes and like there is a lot of differentiation. So we see like metallic individuals way over here and rufipennis individuals with like a ton of variation. And so that would be one of the things to like a cool thing to do in a more controlled way is like, can I like swap their cuticular hydrocarbon profiles and does that change things like mating duration ultimately, you know? So that's my hypothesis is that they're using cuticular hydrocarbons to differentiate different phenotypes and says like, you're different than me. But yeah, that's just my hypothesis. Yeah. Cool. Yeah.
So, for your last part where you talked about the cold responses,
Yeah.
I'm wondering if beetles have the similar machinery as plants do, where you expose them to like cold temperatures, there's a vernalization response essentially. The plants make proteins and stuff that will help them prevent making ice crystals and like just getting their leaves destroyed when there's an actual like frost event.
So like heat shock proteins and stuff.
So, yeah, similar. So do you think that for your tortoise beetles, there maybe some, you may have to look even before a cold event, like either give them like a little bit of a taste to see if there's an upregulation, and then a cold response after.
Oh, yeah. That would be really cool to do. So we did these studies in a way, so we actually measured chill coma recovery twice. So we exposed them the first time, measured that chill coma recovery to the first exposure, and then waited 24 hours and ran it again. And so we do see that the second time, most individuals, but not all, recover faster. So this to me would imply that there is some sort of genetic machinery that does get upregulated from that first exposure that maybe stays upregulated through that next exposure and allows those individuals to recover faster the second time around. Yeah. Can't wait to get the data back. Okay, well thank y'all so much. Oh, sorry. So sorry.
No worries. Thank you for the great presentation. It's super interesting. And before I ask my question, graphical design is awesome.
Thank you.
Are the females of these species able to store and utilize sperm from the males?
Yes.
And the reason I'm asking is I was intrigued by your categorization of some clashes as affecting basically this random pattern of mating. And I'm assuming that, but since by definition the single cross is either restorative or disordered, that there had to be multiple populations for it to work.
Yes, yeah, and so that's exactly what it was. It's basically that female is mating multiply with the males,
Right.
and so because they can store sperm for up to 6 months after a mating event, and the sperm essentially is held in this special sac called the spermatheca, that all of that is, so most of the like random egg clutches are of mixed paternity, basically. Yeah.
That's why I get these proportions that do deviate from
Yeah. It's really cool.
All right, so let's just give Lynette another hand.
Thank you.